Hello everyone, here's Paris from Believers Academy. In short note series, today we are going to cover up the second chapter from 11th standard, biological classification. In this chapter, there are two topics mainly. First is classification, and second is the kingdoms. There are five kingdoms, starting with Monera, Protista, then fungi, plantae, and animalia. We are going to see one by one. In kingdom classification, we are going to see mainly two classification, two kingdom and five kingdom. Two kingdom classification is proposed by Carlos Linnaeus, which includes plantae and animalia. This classification is on the basis of cell wall. Like plantae or plants have cell wall, so they are categorized in one group, and animalia are the group with no cell wall present. Then there is a three kingdom classification, which includes plantae, animalia, and additionally it's have protista. And four kingdom classification includes plantae, animalia, protista, and one more is monera. Now we move to five kingdom classification. This classification was given by R.H. Whittaker in 1969. The year is important. He proposed five kingdom classification, monera, also, then protista, plantae, animalia, and one more additional is fungi. He set some criteria for this classification. Those criteria are important. First is mode of nutrition, then cell structure, then reproduction, body organization, and last is phylogenetic relationship, which means historic or ancestor history of that particular organism to be classified. Let's start with kingdom Monera. The bacteria are the sole member of kingdom Monera. This kingdom or bacteria are categorized as four on the basis of their shapes. First is coccus, that is spherical, bacillus, rod shape, vibrium, comma shape, and last one is spirium, spiral shape. Here are some visual representations of all the four categories of bacteria. Out of this four, only bacillus. Out of this four, only bacillus or rod-shaped bacteria show presence of spore. Now, kingdom Monera is divided into three categories. First is Archaea bacteria, then Mycoplasma, and here it's Eubacteria. We are going to see them one by one. First is Archaebacteria. These bacteria have unique cell wall structure essential for their survival in extreme conditions. Also, with specialized cell wall, they also have specialized cell membrane. These are categorized in three parts. First is methanogens, then halophils, and then thermoacidophils. Methanogens. These bacteria are found in mossy areas, also in gut of ruminant animals like cow. These are responsible for production of methane, that is CH4. Example is methanobacterium. Next is halophils. These bacteria can live in extremely salty areas. The example is halobacterium. Then thermoacidophils. These lives in hot springs or hot uh, water bodies. Example is thermoproteus. Then second part in kingdom Monera, that is eubacterium, which are the true bacteria. These are classified photosynthetic autotrophs, chemosynthetic autotrophs, and heterotrophic bacteria. Okay, so for photosynthetic autotrophs, these are cyanobacteria. unicellular, colloidal or filamentous, freshwater, marine or terrestrial algae. Some of these organisms can fix the atmospheric nitrogen in specialized cells called heterocytes. So if we see this diagram, these cells can fix CO2 and this red color cell can fix nitrogen. These are specialized cells where O2 oxygen cannot enter with. That is the important for fixation of nitrogen. Then chemosynthetic autotrophs. These bacteria are doing oxidation of inorganic substances like nitrate, nitrous and ammonia used to get energy for their ATP production. It's help in recycling the nitrates like nitrogen, phosphorus, iron and sulfur. Example is nitrobacter. Here from this para, the statement based question can be made. Like here they can change the oxidation to reduction and inorganic to organic substances. So you need to remind that. Heterotrophic bacteria, these are the most abundant one, significant decomposers and helpful in production of curd, antibiotics and fixation of nitrogen in leguminous roots. Example is rhizodium. The last part of kingdom Monera is mycoplasma. These are nothing but smallest living cells known and can survive without oxygen. They are aerobic, but if oxygen levels are declined, they still can survive without the proper need of oxygen, supply of oxygen. The size of mycoplasma range from 0.1 mu to 0.3 mu. Yeah, this is important. Then we move towards kingdom Protista. All the single-celled eukaryotes are placed in kingdom Protista. Primarily, they are aquatic and they can reproduction. They can do by sexually or asexually, both ways. This is a kind of question. The first true sexual reproduction process is seen in kingdom Protista because kingdom Monera doesn't have any sexual reproduction process where a zygote is formed. If you see in NCRT says two cells are combining to form zygote. They take the word cell, but they are talking about the gametes. Kingdom Protista is categorized in 5 different types. So first it's starting with protozoans, then cryosophytes, then euglenoids, dinoflagellate, and last one is slime module. Starting with protozoans, they are heterotrophs and lives as predators or parasites. These protozoans are further divided into 4 types. First is amoeboid protozoans, live in freshwater, seawater or moist soil. Example is Amoeba or Echinova. The second one is ciliated protozoans. They are also aquatic for locomotion or moving, active moving organisms with the help of thousands of cilia on the body. If you can see, these are the tiny hair-like structure which help in locomotion for this organism. Third is flagellated protozoans, either free-living or parasitic, having flagella. This long structure is for their locomotion and active movement. Then is sporozoans, the organisms that have infectious spore-like stage. The example is Plasmodium. Next, the classification in kingdom Protista is chrysophytes. This includes diatoms and golden algae, which is dismids. This name golden algae is just given to dismids because of color only. They are freshwater living or marine. Here's the chart of difference between diatoms and dismids. Talking about cell wall, diatom cell wall is made up of silica, which is non-degradable substance, whereas dismids cell wall is made of cellulose and pectin. Habitat, they are freshwater and saltwater, but dismids are just primarily saltwater. The shape of diatoms are box-like or disc-like shape, whereas here we can see variety of shapes, including elongated and star-shaped. This table you have to remember because direct question can come for the atoms. Next is urinoids. The majority are freshwater living organisms found in stagnant water, which is still water, like lake, in lakes or in ponds. These are photosynthetic in presence of sunlight and predatory in absence of sunlight. If we provide sunlight, they do photosynthesis and make their own food. If the sunlight is absent, they become predatory. Then comes dinoflagellates. Then come dinoflagellates. These are marine and photosynthetic. They appear in multiple colors like yellow, green, brown, blue, or red. We are specifically talking about the red here. The the red dinoflagellate can undergo rapid multiplication and can make C appears red. It is called as this is called as red tides, which are made by this red dinoflagellates. You can see for their locomotion, they have this long filament, and you can see the structure over here. Then last part in kingdom Protista is slime modules. These are these are saprophytic protistas moving along decaying twigs and leaves. Then here's the interesting part from plasmodium, which grows and spreads under favorable conditions. Let's talk about this. If this is a decaying substance, then the slime modules get grows and like spread on this substance in favorable conditions. In upper side we can see as like this and with multinuclear structure. If if so, if the conditions become unfavorable, these slime modules, instead of spreading, what they do, they start to make a small tiny projections, and these small tiny projection holds the spore for their growth. When conditions become favorable, this outer covering is then cut and these spores get come out. They combined, form a new zygote, and then it grows to form another slime mold. The interesting part over here is these plasmodium do not have its cell wall. Its cell wall is absent, but this haploid spore have cell wall, which is cellulosic cell wall. These are the direct questions for your NEET, that plasmodium do not have cell wall, but spore contains cell wall in haploid condition.